Hi guys, in this video we'll be starting with enzymes and we'll be covering characteristics and naming of enzymes. So let's get started. So first we need to understand the meaning of a term catalyst. Now a catalyst basically is any substance that changes the rate of a reaction without being itself affected or without being itself changed at the end of the reaction. Okay, basically they don't participate in the reaction. Okay, so enzymes are specific types of catalysts. They are biological catalysts and they alter the rate of a chemical reaction and they don't participate in the reaction. Okay, this is the complete definition of an enzyme. Okay, so basically in some cases, for example, when organisms such as fungi and flies digest their food externally, okay, they control the rate of reactions happening outside the bodies of organisms. So most of the reactions that provide cells with energy and produce new biological material would take place very slowly at the temperature and pH of living things, and it would be too slow for life to exist. So enzymes make life possible by speeding up the chemical reactions in cells without changing the conditions in the cytoplasm. Okay, so let's look at the characteristics of enzymes. So the first one is basic. It's just that they alter the rate of a chemical reaction, and usually they speed up the rate. Okay, then they remain unchanged at the end of a reaction. Okay, they all are globular proteins in nature and hence they are soluble in water. Okay, and they all are specific in nature. They all catalyze reversible reactions only, and they are used in minute amounts. They are used in very small amounts, and they work best at optimum pH and temperature. Okay, and the rate is also affected by changes in substrate and enzyme concentration. Okay, concentrations. Okay, moving on to the next property. Another important property, and this is very important, that the rate of the enzymatic reaction, okay, is reduced by specific inhibitors. Is reduced okay, we will come to this later. And the final point is that many of the enzymes, they need coenzymes, okay, for their proper working. And what are coenzymes? Coenzymes are basically non-protein complexes, and this is the final point in the characteristics of enzymes. Okay, now let's come to the naming of enzymes and different types of names given to an enzyme. So basically in medicine, in cellular and genetic research, and in industries that use biotechnology, It is very important to be able to refer to the action of specific enzymes. Okay, to do this, okay, to accomplish this, we need to understand how enzymes are named. Okay, so many of the enzymes found in animals and plants work inside the cells. The enzymes that catalyze reactions inside the cells, they are known as intracellular enzymes. Okay. And the enzymes that catalyze reactions outside the cells are called extracellular enzymes. So these are the two major groups in which we divide the enzymes. Okay. Examples of intracellular enzymes include DNA polymerase, DNA ligase, okay, and examples of extracellular enzymes include the digestive enzymes and lysozyme, the enzyme in tears, okay. These all are included in extracellular enzymes. Moving on, okay. So most enzymes, okay, they are named by a relatively short recommended name, which is often the name of the molecule that the enzymes worked on. Okay, the substrate, okay, on which the enzyme works on, and it's completed by assigning ASE on the end. Okay, for example, we have creatine kinase, sorry. Okay, the second naming system is the longest systematic name. Okay, longer systematic name, and it describes the type of reaction. Okay, type of reaction being catalyzed. So for example, you can have ATP, okay, creatine phosphotransferase. Okay, and the third type of naming is basically a classification number. So for example, this is just an example. Okay, example of a classification number can be like this. Okay, we also name enzymes according to the specific type of reactions that they perform, or the particular function. Okay, the particular function that they perform, or the reactions that they perform. Okay, and also in this we add ASE in the end. So for example, hydrolysis. Okay, let's take an example. Hydrolysis. Okay, so all the enzymes involved in hydrolysis will be called hydrolases. Okay, hydrolysis. And for joining proteins, we have ligase. Okay, that's for joining proteins. This is for hydrolysis reactions, and we also have polymerization reactions, right? So for polymerization reactions, okay, the enzymes are generally called polymerase. Okay, so this, and similarly, according to different reactions, you can name these enzymes. So there are quite a number of ways to name enzymes. Okay, so this is all for this video. Thank you for watching. We'll meet in the next video. Till then, take care.